Hello everyone. From now on, we will present a study on the factors that slow down the decay of meat. In chapter 1, we will talk about the motivation for selecting this topic and associated theories. In chapter 2, we will talk about experimental purpose and process and result. In chapter 3, we will talk about the consideration and realization and conclusion.
Let's start with our topic selection. In the hot summer, if you are a storage of food, food poisoning will breed, and we've decided to explore raw meat, which is a major cause. Meat is said to be decay in about 5 hours at room temperature, and we choose this topic to increase the storage time. The following is a presentation on the relevant theory. First, let's talk about decay. Decay is the phenomenon in which organisms are deodorized and decomposed by microbes, mainly protein breaks down. When meat loss occurs, the color of the meat changes and the taste of alkaline can be seen through pH. The following are factors that can prevent decay. By making oxygen cut off and pH less than 3, the effect is as follows. We use materials to suppress the factor that cause decay in this experiment. First, salt stops microbial activity by osmosis, which causes dehydration or slows movement. Next, the peroxide sugar solution absorbs moisture and thus inhibits microbial activity. Lactobacillus develops microbes to prevent germs from proliferating. Vitamin C prevents oxidation.
Next, let's find out about the purpose of the experiment. Knowing that the rate of decay in meat stored at room temperature is faster than the rate of decay in refrigerated meat, we were experimenting to find and devise ways to solve factors that could slow down the rate of decay at room temperature. We first hypothesized that muscle mass and fat would be different in degree of decay. Later, a group of experiments were compared with materials that could exclude high temperatures and high humidity conditions, as well as whether they were in contact with oxygen, which is responsible for the cause of decay. The observation cycle was incorrectly set and results were not obtained. Therefore, the second test was conducted to identify the problem in the first test and to find a point to compensate. The second phase of the experiments is like this. First, divide each meat into a certain size and mass. Second, set up experimental and control groups and apply the solution we thought to meat. Third, divide the observation cycle and observe it. Fourth, measure and compare the MBL kit and pH test paper to find out the result of the experiment. Lastly, recognize the difference in decay, though bacterial culture. This process leads to the experiment.
Then, this is the first experiment result. It was planned to be observed up to August 9th with experimental preparation in August 5th, but could not be measured as decaying was completed in August 6th. 1 hour later, it was visually confirmed that there was fluid around the meat. We could observe the process in which liquids are being produced in salt and vitamin C, except for oversaturated sugar solution and vinegar. And 2 hours later, almost all meat was liquid and began to stink. 3 hours later, the smell of meat was stronger around us. 4 hours later, with a tremendous stench, the surrounding flies were twisted and lot a lot. 5 hours later, there was little difference between 4 o'clock, but the bright red meat gradually lost its color, and the volume of liquid increased and became high. This is the pork neck's pH. This is the pork belly's pH.
We originally planned to observe it every 24 hours through microbial culture and pH measurement. However, from August 5th to August 9th, when it was supposed to be observed, decay was quickly carried out and failed due to high temperatures of 34.3 degrees on average and humid conditions caused by typhoons. So, we prepared the second experiment on August 17th. The observation cycle was reduced to 1 hour and the experiment was carried out using air conditioner. This RNE was a great experience for us to explore and discuss our topic by selecting topics that we were interested in and experimenting with. And we realized that even if the experiment fails, we can continue to do it and produce results. In conclusion, the first experiment failed. In the second experiment, we were able to identify the factors affecting decay as a result. Further research opinions have been established, and the preservatives will be produced as a further research.
Thank you for listening to our presentation so far.